Now on this page of our notes, we're going to get into some basics related to equations, reactants, products, what we mean by equilibrium, and then we're going to talk about acids and bases. We're going through some very simple chemistry concepts, and we're just going to talk about what you need to know for the AP Biology exam. You're probably used to equations looking like this. So we see some pluses and we have some arrows there. You should know that anything that's on this side of the arrow is considered the reactant, and what it is pointing at is going to be considered the product. Now I want to make sure that you understand that reactions can happen in two different directions. If we have an arrow showing these reactants turning into the products, then we say this is the forward reaction. But water can also break back down into its reactants. And so sometimes you're going to see equations and you're going to see arrows that look like this. And this is just simply to show you that we can have forward reactions, but we can also have the reverse reaction. I'm writing reverse there. And that's when the products are going to break back down into the reactants. Now that we've said that reactions can happen in the forward direction or the reverse direction, the direction of this reaction usually depends on the concentration of the reactants and products. If you have more reactants, then you're going to get this forward reaction, and these reactants are going to join together to form these products. If you have a large amount of the products and not many of the reactants, then we see more of the products will then break back down into the reactants. I do want to talk about this next thing, the conservation of mass, which states that mass or matter can never be created nor destroyed. And whenever you see an equation, you're probably going to see it's balanced. Hopefully you recognize this equation. This is the equation for photosynthesis. If you do not have it memorized by now from pre-AP, then you need to start memorizing it because you're going to see it a lot in AP. The problem with this equation that you see right here, we have CO2 plus water, and then energy from light is making glucose and oxygen. The problem is the equation is not balanced. So whenever you see the equation for photosynthesis, you're going to see coefficients in front of these different reactants and products so that we have a balanced equation. So let's go ahead and indicate what that correct equation for photosynthesis is going to look like. We actually are going to have to use 6 carbon dioxide molecules and 6 water molecules. And that's only going to give us 1 molecule of sugar, and it's going to release 6 molecules of oxygen. Let me go through what I mean by balanced and how this relates to the conservation of mass. If you take a look at our reactants over here, and let's look at the oxygens. Here I have 2 oxygens. There's 1 oxygen. So I have 3 oxygens. I have 3 oxygens on this side of the equation. But on this side of the equation, you can see that I have 6, 7, 8. I have 8 oxygens. If this is really what was happening during photosynthesis, then somehow there has been the creation of 5 oxygen atoms. And we know that cannot happen. Atoms do not form spontaneously with just protons, neutrons, and electrons coming together. So matter can never be created nor destroyed. It just changes forms. If we want to end up with, in this case, we have 12 oxygens over here plus 6 oxygens here. So we have 18 oxygens over here. In order for 18 oxygens to come out, we had to put 18 oxygen atoms in. And whenever I added these coefficients before these reactants, now I have, there's 2 times 6, so I have 12 oxygens, so I have 12 O's there, and I have 6 times 1, that gives me 6, so I have 18 over here and 18 over here, so we have a balanced equation. Now that we've just talked about ions, I want to go ahead and tackle acids and bases and what you have to know for the AP exam. An acid is any substance that when you add it to a solution, it will increase the H plus concentration. Now, H plus really just simply means a proton. Because if you have a hydrogen atom, it's got 1 proton. So I'm going to write hydrogen atom. It has 1 proton and 1 electron. And the only way for this hydrogen atom to have a plus charge is if it has donated, it has gotten rid of its electron. And then if it has 1 proton but no electron, it's going to have a plus 1 charge. So really, when we write H+, I need you to know we're just talking about a single proton. So I can call this the H+ ion, I can call it a proton, or sometimes you'll hear me say hydrogen ion concentration. So back to acid over here. Something is considered an acid if I add it to a solution. I'm going to increase the hydrogen ion concentration in that solution. So let's take a look at what I mean by that. Here we have hydrochloric acid, and the hydrogen atom is bound to this chloride atom by an ionic bond. So let's indicate holding those together is an ionic bond. And we already established that ionic bonds are weaker bonds. So if I take hydrochloric acid and I put it in water, it's going to disassociate. That means it's going to break apart. And it's going to break apart into hydrogen ions and chloride ions. And the reason why is because hydrogen likes to give up, completely donate an electron to this chloride atom. So this chloride atom is going to have an extra electron, so it's going to have a -1 charge, whereas this hydrogen atom has given up an electron, so it's going to have a +1 charge because it has one more proton than it does electrons. Let's take a look at what that looks like. Here I have a beaker of hydrochloric acid, and here I have a beaker of pure water. As I indicated up here, water is breaking down and it's reforming. So that's why you see this hydroxide ion here, this hydrogen ion here, because they used to be together as a water molecule, but they can break apart and then they can reform back together to form water. So we get both the forward and the reverse reaction. We have simply a beaker of water. If I take some hydrochloric acid and I add it to this beaker of water, then I already said that the hydrochloric acid is going to break up into ions. And you can see now all of these hydrogen ions that are in the solution. So adding hydrochloric acid increased the hydrogen ion concentration of this solution. That's why hydrochloric acid is considered an acid. Let's take a look at a base. A base is any substance that whenever you add it to a solution, it's going to decrease the hydrogen ion concentration. Here's some examples of some bases. So I have sodium hydroxide. Whenever I take sodium hydroxide and it's bonded together ionically and I add it to water, what happens is it disassociates and we get sodium and we get this part is called the hydroxide ion, and it's going to have a negative charge to it. So let's go ahead and take a look at what happens here. Here's my beaker of sodium hydroxide. I add it to a beaker of water and watch what happens. The sodium hydroxide splits apart into ions. And what can happen now is this hydroxide ion is going to react with these hydrogen ions. And you can see that whenever they react together, so let's cause these two to react together. Now I no longer have these free-floating hydrogen ions. So by adding sodium hydroxide to this beaker, I'm actually lowering the amount of hydrogen ions that are in that solution. And by definition, that is what a base does. It's a substance that when added to a solution, it decreases the hydrogen ion concentration. Now let's talk about the pH scale and what you have to understand about that. This pH scale is actually measuring the hydrogen ion concentration of a solution. You need to know that right here in the middle, 7 is a solution that is completely neutral. Less than 7 and we're getting acidic. More than 7 and we're getting basic, or we can say alkaline. Now, this is a confusing thing about the pH scale and what you have to understand about it in relation to the hydrogen ion concentration. There's an inverse relationship between pH number and hydrogen ion concentration. What I mean by inverse relationship is as the number gets lower, so as the pH number gets lower, so as I go from 7 to 6 to 5 to 4, so as the pH number gets lower, my H plus concentration gets higher. So to help you remember this, let's draw a beaker here. And the solutions that have a low pH are going to have a ton of free-floating hydrogen ions. So the more hydrogen ions we have in a solution, the lower the pH is going to be. So the lower the pH, the higher the hydrogen ion concentration. The higher the pH, so as we get up to 8, 9, 10, 11, 12, the lower the hydrogen ion concentration. So let's just draw a beaker here, and let's just put one hydrogen ion in that solution so that you understand high pH means lower concentration of hydrogen ions. Let's work some grid-in problems that you're going to have to be able to do related to AP biology. One thing you're going to have to do is if you are given the hydrogen ion concentration, you have to tell me what is the pH of the solution. And pH is always going to be between a number 0 and 14. We're not going to go higher than 14. This is a very easy question. All you have to do to answer this is you have to look right there, and that tells you the pH of the solution. So if you have a solution and the concentration of hydrogen ions is 1 times 10 to the negative third, then the pH of the solution is going to be 3. Next one, the hydrogen ion concentration is 1 times 10 to the negative 8th. What is the pH of the solution? It is 8. Now, let's talk a little bit about scientific notation. What does it mean if you have a number that's 1 times 10 to the negative third? Well, let me turn that into a decimal for you. I'm going to put this one, I'm going to write 1, and I'm going to put a decimal there. So we have the number 1, and it says 10 times the negative 3. Negative 3 means you're going to move the decimal, and you're going to move in this direction 3 spots. 1, 2, 3. I'm going to put my decimal right there. Fill in my spots with 0. So 1 times 10 to the negative 3 is the exact same thing as this decimal that I just wrote. If I wanted to write this in decimal form, I'm going to write my 1. I'm going to put it over here so I have room. And it's 10 to the negative 8. So we got to move it places to the right because we're going to make a really small number here. So 1, 2, 3, 4, 5, 6, 7, 8. Ran out of room. Put my decimal there. So I have 1, 2, 3, 4, 5, 6, 7 zeros before that 1. Let's take a look at number 3. It says the solution has a pH of 10. What is the concentration of hydrogen ions of the solution in scientific notation? Here's scientific notation. So we just need to write 1 times 10 to the negative 10. Because remember, pH just simply equals what we have there for that superscript on the 10. Number 4 says a solution has a pH of 4. What is the concentration of hydrogen ions in a decimal? So if the pH is 4, let's write scientific notation. 1 times 10 to the negative 4, but in decimal form. Let's put our 1 there and move it 4 back. 1, 2, 3, 4. Put your decimal there and fill in with zeros. So 0.0001 is the answer to that question. So take a look at number 5. It says the hydrogen ion concentration of a solution is this number as a decimal. What is the pH of the solution? Now we're going to have to move the decimal the other way and get it back into scientific notation. So let's move. We got 1, 2, 3, 4, 5, 6. So we are 1, and it was times 10 to the negative 6. You can go back the other way if you want to make sure you did it correctly. So start here with the decimal here and go 1, 2, 3, 4, 5, and 6. So the pH of our solution is going to be 6. Here's another type of problem that could be a grid-in. You need to know that if we change by 1 on the pH scale, that actually means there's a 10 times change in the hydrogen ion concentration. So let's take a look at these related grid-in questions. The pH of a solution changes from 13 to 12. The concentration of the hydrogen ions, let's take a second to think about it. Is the hydrogen ion concentration increasing or decreasing if we're going from 13 to 12? If we're going from 13 to 12, we're going this way on the pH scale. And remember, we drew that beaker and we put tons of hydrogen ions in it. So the concentration of hydrogen ions increases, and we've only moved 1 on the pH scale, 12 to 13, so it's by 10 times. Taking a look at number 2. It says the pH of a solution changed from 3 to 5. The concentration of hydrogen ions. So now we're moving this way. And remember, we ended up with a beaker over here, and we just put 1 little hydrogen ion in that beaker. So we know that as you become more basic, then the concentration of hydrogen ions is decreasing. So let's put decreases. Now, let's go ahead and let's write our numbers out. This is the easiest way to do it. This is pretty foolproof. Remember, a change of 1 is 10. And then a change of another 1 is 10 as well. We're not going to add these together. You have to multiply. So my change in hydrogen ion concentration, it's decreasing by 100 times. 10 times 10 gives you 100. Let's do the next one. It says the pH of a solution is 4. The hydrogen ion concentration increased, that's important, by 1,000 times. What is the new pH of the solution? So let's start at 4. We're increasing the hydrogen ion concentration. So we're going this way on the pH scale. We're going to end up making it more acidic. Okay, so let's take it down to a 3. That's going to be a change of 10. Let's take it down to a 2. That's going to be a change of 10. We're at 100 now, so now we're going to have to go down to 1. That's a change of 10. 10 times 10 times 10 is going to give us that 1,000. So our new pH of the solution, our answer, is 1. On your formula sheet, you actually have an equation that shows you how to calculate pH. One thing I want you to know is that whenever you're taking the AP exam, you are only allowed to use a 4-function calculator, and it does not have a log button. So basically, if you can do these problems that we just went through, you are technically using this formula to answer those questions. But you won't really use that formula per se because you just don't have a log button on a 4-function calculator. So if you have this memorized, that a change in 1 pH changes the hydrogen ion concentration by 10, then basically you are using that equation. Now we're going to take a look at acids and bases and something called buffers, and we're going to relate it to a biological concept. Now, we have stated many times in pre-AP biology that organisms must maintain homeostasis. So that means that organisms are trying to maintain a constant, non-changing internal environment. So you're trying to maintain a certain level of pH, a certain temperature, a certain concentration of water in your system. Like I just indicated, organisms, they have to really carefully regulate pH. And you have a system in place, which is known as a buffer system. And a buffer system helps you maintain a very narrow range of pH. As a matter of fact, your blood in your body stays anywhere between a 7.3 and a 7.4 on that pH scale. So it's a very narrow range. A pH buffer is going to cause a solution to resist a change in pH. So here's what I mean by that. Let's say you have a beaker of water and you add a buffer to it. So now let's add an acid to that water. If that water initially had a pH of 7, even though I added an acid to it, it's going to stay around a 7 because I put a buffer in it. Same thing if I have water in a beaker with a buffer, and let's say the pH starts out at 7 again. If I add a base, the pH isn't going to change to like a 10, it's going to stay at a 7 because of this buffer system. Well, the key buffer system that you need to note and be able to recall is the carbonic acid bicarbonate ion buffer system. If you remember back to a demo that you probably did in your pre-AP class, and that's where, so remember we added some water to a beaker, and then we put a straw in there, and if you remember, you blew bubbles. And basically you're exhaling CO2, so you bubbled CO2 into the water. Now, in addition, we put an acid-base indicator. Remember, it was called BTB, bromothymol blue. When I added it to the water initially, it turned blue. But as you blew bubbles, that water that was blue, it turns yellow because you're making carbonic acid because you're adding CO2 to water. And that's what I'm showing you here with this equation. So when you add water and CO2, you make carbonic acid. Now, carbonic acid can dissociate. So hopefully you understand what this equation is showing you. It can break down into this, what's called the bicarbonate ion, whenever an H plus dissociates from that bicarbonate ion. And there's that H plus right there. When I talk about carbonic acid, this is the chemical equation for carbonic acid, and this is the equation for that bicarbonate ion, which comes from the breakdown of carbonic acid. Now here's how this buffer system works. And so I'm gonna define it for you, and then I'm gonna show you in pictures how this buffer system helps you maintain pH. Whenever your blood becomes basic, too basic, then what happens is carbonic acid breaks down. So let's show that, H2CO3. This forward reaction results. You end up with the bicarbonate ion, but look what you end up with. You end up with those hydrogen ions being donated into the solution. So if your blood is becoming basic, that means that the hydrogen ion concentration was decreasing. Well, this carbonic acid can now donate hydrogen ions to the solution and bring your pH where it was normally. If your blood is becoming too acidic, that means that you have an excess of hydrogen ions. You need to get rid of them. What happens is this bicarbonate ion will accept it. And so what actually happens is the reverse, is this bicarbonate ion is going to bind to all those excess hydrogen ions, and it's going to form carbonic acid. And it's going to take those hydrogen ions out of solution. So let's take a look at that in regards to a picture here. So here I have a solution that has carbonic acid in it. Remember, carbonic acid can break down into that bicarbonate ion and hydrogen ions. We're going to add an acid to this system that has this buffer in it. Now, first though, I want you to note how many hydrogen ions we start with. So in this situation, I have 4. And because we have this buffer system in the solution, we're going to stay at 4, even if we add an acid. So I'm going to add some hydrochloric acid to this system. So when that happens, the hydrochloric acid, it breaks up into ions. So I'm going to put some of those ions now in that solution. The pH has changed because we've got 1, 2, 3, 4, we have 5, 6 hydrogen ions now. So the solution right now has become acidic, but remember what the buffer does. The buffer is going to keep our pH where it was initially. Here's what happens in this situation. Since the solution becomes more acidic, what happens is these bicarbonate ions will bind to these excess hydrogen ions. So we can bind this one right here to this one. And now if you look back and count our hydrogen ion, we have 1, 2, 3, 4. And so our pH did not change. Our pH does not change if you have a buffer in place. Again, here we have a solution with a buffer in place, and we're going to add a base to it. But again, we're going to keep the hydrogen ion concentration the same. So we're starting off with 4. Once again. So let's add a base. So let's add some sodium hydroxide. And we know that sodium hydroxide is going to break apart into sodium ions and hydroxide ions. So let's put a few in there. Okay. What's going to happen when you add a base? Remember, a base actually decreases hydrogen ion concentration. And it does that because these hydroxide ions are going to go bind to some of these hydrogen ions to form water. Well, if we look at the scenario now, we have decreased hydrogen ion concentration. The solution is more basic. There's less hydrogen ions. There's only 2 of them. Well, the buffer, again, is going to bring pH back to what it initially was. And so that's where this carbonic acid comes into play. It's going to disassociate, so we're going to see this forward reaction that I was talking about before. And this carbonic acid is going to break down into hydrogen ions and, sorry, I'm writing it wrong, and this bicarbonate ion, HCO3 negative. And here's a carbonic acid, so let's break it down. It's going to release those hydrogen ions and form that bicarbonate ion, HCO3 negative. And let's kind of cross through these because now these hydrogen ions are tied up as water molecules. So now let's look and count how many hydrogen ions we have. We have 1, 2, 3, and 4. Whenever the solution became basic and more hydrogen ions were needed in the solution, those hydrogen ions came from the dissociation of that carbonic acid. So we say that carbonic acid donated hydrogen ions when needed. And that's what I have summarized for you in this box. So if the blood becomes too alkaline or too basic due to these hydroxide ions binding to these hydrogen ions, decreasing hydrogen ion concentration, that's when the carbonic acid is going to donate hydrogen ions. But again, if you have a buffer in place, that pH of a solution will not change. This buffer system in your blood offers an advantage because it allows you to keep pH at a certain range with very little fluctuation. Now, the second thing that we're going to take a look at is acids and bases and pH and what's called the Bohr effect or the Bohr shift. We've mentioned that proteins in your body have to maintain a specific shape to function. And the shape of proteins is affected by the pH of the solution. If you remember that we talked about like proteins like enzymes, they have an optimal pH. And if you get too far from that pH, the enzyme won't function. The enzyme will become denatured. So you have like a denatured protein here at a low pH, and you have a denatured protein at this higher pH. And so the shape is being changed due to the pH of the solution. Our bodies use that as an advantage in some situations. So we're going to talk about a protein that carries your oxygen around your body, and that protein is in your red blood cells, and that protein is called hemoglobin. And we know it's a protein because it ends in IN. So hemoglobin is a protein. If hemoglobin is in a solution with 1 pH, it's going to have a shape that's different from whenever it's in a solution with a different pH. Crash course in the respiratory system is blood is sent to your lungs, and your blood is going to pick up oxygen from your lungs. So specifically, the oxygen moves from your lungs into your blood, and it binds to hemoglobin. And that blood's going to travel to other areas of the body, to different tissues, and those red blood cells with that hemoglobin is going to release oxygen into your tissues. Now, how is it that hemoglobin can bind to oxygen in one area but actually release it somewhere else? Well, it has to do with there's a difference in pH between your lungs and other tissues of the body. So I'm actually going to start over here, and I'm going to write body tissues, because this box summarizes what happens in your body tissues and how pH is different there. You know, we talk about your cells. They're taking in oxygen and they're making CO2 in the process of respiration. And we said that if you have CO2 and it comes in contact with water, it makes carbonic acid. In areas of your body where you're using a lot of oxygen and making a lot of CO2, those areas become acidic. Now, whenever the red blood cells travel into those areas that are more acidic, that hemoglobin is going to have a shape change. And that shape change is going to cause that hemoglobin to actually release oxygen. It can't bind to it as well because it's had a shape change. So in these acidic environments, hemoglobin is releasing oxygen because it has less of an affinity for oxygen. That means it's less likely to bind to it. In acidic areas of the body where there's a lot of CO2 being produced, those areas need a lot of oxygen, and that hemoglobin will release more oxygen to those areas that have a high oxygen demand. So let's talk about at the lungs. The lungs, the blood is actually releasing CO2 from the blood into the lungs. And so your blood is not as acidic. It's more basic. Whenever the hemoglobin experiences an environment that's less acidic, it has a shape change, which actually allows it to bind to more oxygen. So it has a greater affinity for oxygen, which means it'll bind tighter, it'll bind to more oxygens in areas that are less acidic, such as your lungs. So basically what this is indicating is that this is the reason why oxygen will bind to hemoglobin whenever the red blood cells are in the lungs, but the red blood cells will actually release oxygen to your tissues, because at your lungs, it is less acidic. In your tissues, it's more acidic. Write that up here. It's more acidic. And that change in pH is changing the hemoglobin, HB, that hemoglobin, changing its shape and causing it to actually release some of that oxygen.